Hello everyone, this is Creative Biogene. Today we will tell about analytical testing services. Analytical testing service is a deep analysis of samples through biological, chemical and physical method or with high-tech instruments. Utilizing analytical testing, it is accurate and rapid to find the trace contamination of products, including microbes, virus, adventitious cell and reagents. Contamination is considered as the main problem in the manufacturing process of biological and clinic products. These main contents include host cell protein assay, residual DNA testing, next-generation sequencing, and transmission electron microscopy. Host cell proteins are proteins produced or encoded by the host organisms that are used to produce biological products. Biological products, especially therapeutic proteins, are usually produced by genetically modified prokaryotic or eukaryotic cells. During the biological product's production, host cells also co-produce proteins related to the normal cell functions such as cell growth, proliferation, survival, gene transcription, protein synthesis etc., which become a major part of process-related impurities. Host cell proteins are critical to drug safety, efficacy and regulatory qualification. Incomplete detection in biotherapeutics development can lead to product degradation, product instability and adverse immunogenic reactions in patients. Therefore, it is a regulatory requirement to monitor the removal of host cell proteins and assay the residual host cell proteins in drug product during bioprocess development. The methods used for identification and quantification of host cell proteins can be classified as either specific methods, such as ELISA and Western blot, or non-specific methods, such as electrophoresis and mass spectrum. Among these, ELISA, electrophoresis and mass spectrum are the most common techniques, and sometimes they are used in combination for detailed characterization. Creative Biogene can offer various methods to detect and assay the amounts of residual host cell proteins, as well as providing a variety of ELISA-based host cell proteins assay kits for your research. Biopharmaceutical products, such as therapeutic proteins and vaccines, are produced by fermentation using either bacterial or eukaryotic cells. The cells used to produce biopharmaceuticals can be sources of a range of complex, heterogeneous, and potentially unsafe impurities, and host cell DNA is among these. The residual host cell DNA may result in tumors or adverse reactions. Besides, cells used to produce biopharmaceuticals may possibly carry viruses or harbor harmful nucleic acid, and the residual DNA in a given biopharmaceutical product may be infectious. Therefore, some regulatory agencies have allowed a target of 100 pg or less of residual DNA per dose in biopharmaceuticals, and levels up to 10 nanograms of residual DNA per dose may be considered, depending on the source of the residual DNA and the product's route. Currently, the most commonly used method of detecting and quantitating residual DNA is quantitative PCR. Creative Biogene can offer highly sensitive methods to detect and quantify minute amounts of residual host cell DNA, as well as providing a variety of qPCR-based residual DNA quantification kit for your research. Next-generation sequencing, a massively parallel or deep sequencing technique, has revolutionized genomic research. Compare with the first-generation sequencing, the next-generation sequencing is characterized by high accuracy, fast speed and low cost. So far, there are many NGS platforms involving different sequencing technologies. Although these platforms differ substantially in terms of their engineering, sequencing chemistry, output, accuracy and cost, all of NGS platforms perform sequencing of millions of small fragments of DNA in parallel. As a high-throughput sequencing technique, NGS applies to genome sequencing, genome resequencing, epigenome characterization, DNA-protein interactions and transcriptome profiling. Now, NGS becomes an efficient and effective tool for biosafety testing. Based on the high accuracy and in-depth analysis of nucleic acid, NGS is used for multiple applications including pathogen safety testing and genetic characterization. Creative Biogene brings pharmaceutical and biotech companies next-generation sequencing service. We provide a complete next-generation sequencing service from sample preparation and library preparation through sequencing to data analysis. The next-generation sequencing can be applied for adventitious agent detection, virus seed stock, bacterial cell line identification, cell substrate genetic stability testing, and expression vector identification. Transmission electron microscopy is a microscopy technique in which microscopes use electrons instead of light as an emission source. Comparing with light microscopy, transmission electron microscopy gets a resolution 1,000 times owing to its electron source with much lower wavelength. Using transmission electron microscopy, it is possible to capture fine detail, even as small as single column of atoms. Transmission electron microscopy produces high-resolution and detailed images from which researchers can get more information on element and compound structure. As a major and powerful analytical method, transmission electron microscopy plays an important role in chemical, physical, and biological sciences. Now, transmission electron microscopy is considered as a tool for biosafety testing and applied for viral particles detection, microbial contamination identification, material analysis, and so on. Sample preparation is a crucial step before a transmission electron microscopy analysis, which includes dehydration, spotter coating of non-conductive materials, cryofixation, sectioning, and staining. Creative Biogene offers an expertise transmission electron microscopy service for clients. Our expertise in transmission electron microscopy-based biosafety service allows us to provide full-service GLP electron microscopy evaluations, from sample preparation through image acquirement to data analysis. The advantages of our analytical testing services include leading-edge technology and expertise, affordable costs and cost-effective prices, reliable and high-quality data analysis, and experienced scientist team. If you have any questions, please feel free to contact us. We look forward to working with you in the future. Thank you for watching.